will introduce you. So our next speaker, it's a short talk, is Ashwin Narayan, okay, from the MIT, and he's going to talk about density-preserving visualizations of single cells, and he's all set. So.
Got to lower this. Can everyone hear me? Yeah, good, okay. All right, hi everyone. So I'm going to talk about some work that we've been doing towards developing what we're calling density-aware algorithms for visualizing single cells. And I apologize for subjecting you to another review of TSNE, but here we go. So basically the goal of TSNE is, as you all know by now, to embed high-dimensional data into 2 dimensions in a way that preserves relationships and/or structure. And essentially, the way it works is by trying to preserve local neighborhoods in an embedding without really paying attention to long distances. And for a concrete example, suppose we have this toy data set here. So the embedding of this point highlighted in orange is going to pretty much depend on the embeddings of the 3 points close to it, and not at all on this point that's pretty far away. And similarly for that other orange point. Now, you might notice pretty quickly that the length scales of the neighborhoods for these 2 points are actually quite different. And one of the very clever things that TSNE does is automatically determine the length scale for a particular point in the embedding. And then it normalizes by that length scale. The problem with that is you immediately lose all density information after you do this normalization. And again, a concrete example. So suppose this is just a scaled up version of the previous data set where now you have 2 Gaussians, both with the same number of points, but one which clearly has a much larger variance than the other. And it's a well-known result that when you do TSNE, both the clusters that emerge will look exactly the same. And this comes from a blog called Misread TSNE. It's a really good blog post, and I highly recommend everyone who uses TSNE to read it first. And so while this is an obvious distortion, it actually becomes more nefarious if, for example, you have 2 clusters that have a big overlap region where, say, that overlap region has a higher density than the areas around it. You get these weird distortions. So that's why our goal is to come up with an algorithm that actually preserves some notion of the original density in the embedding. So before we can preserve density, we first need to have a good way to measure it. And we want our measure to be smooth so that we can actually optimize it using some kind of gradient-based algorithm. We want it to be robust, so it doesn't necessarily depend much on parameter choices or on it shouldn't even be specific to just TSNE. And we want it to be intuitive so that it actually matches what our internal or intuitive notion of density is. And to that end, we are presenting this idea called the local radius. And it's very simple. It's basically a smooth version of taking the average distance to a point's nearest neighbors. So the local radius at a point is more or less the average distance to its nearest neighbors. And again, as a concrete example, we're going back to our previous data set. The local radius of this first point is going to be the radius of this circle, which kind of is the average radius of those 3 points around it. And this other point is going to have a bigger local radius. And so a successful density preserving algorithm, a point in the original space with a large local radius will also have a large local radius in the embedded space. And in order to, when we think about optimizing this, the first thing to note is that since the volume of a space increases as the power of the radius, what we're really hoping for is a nice power law relationship between the original and the embedded local radii. And so to that end, what we're going to actually try to do is maximize the correlation coefficient of the log of the original and embedded local radii. And like I said before, since we have a smooth measurement, we can actually just do this by gradient ascent. And as we heard in the previous talk, TSNE optimizes its own objective function via gradient descent, so we can also basically add a density term to that gradient. And so this is the only equation in my talk. This is the objective function that is going to be maximized. It's basically just the covariance of the original and embedded radii normalized by their standard deviations. And now we can basically take a look at some data sets. So first, just starting with a nice, easy simulated data set. Here we have 10 different Gaussians whose means are basically translated and their variances are getting bigger. So we have this big overlap region. And essentially you can think of this as maybe representing a cell differentiating, but not differentiating super quickly. So these overlaps are present. And when you run t-SNE on it, you see what I was talking about before is these like distortions. Does this laser work? No. You see these distortions in the interfaces between the intersections of the different colors. And you also don't see the expansion into expression space that I think is pretty important for differentiation. And now when you run our algorithm, the distortions at the intersections go away, and you also start to see the expansion that we're looking for. So I have another example where the red is not as close to the yellow. So one of the bad things about t-SNE is it's not, different runs will produce slightly different results. And as we know, real data is obviously much trickier than simulated data. So here I have a t-SNE plot of, I think, 20,000 mouse brain cells where each color represents a different cell type. And when you run the density version of t-SNE on it, qualitatively you get very similar things. So the clusters are still as well separated as you would hope for with the t-SNE plot. But there are some different shapes and I have a poster upstairs with which you can actually look at it closer and interrogate this a little bit more. But the thing I want to show you most is that when you actually regress the original and the embedded density, in the original t-SNE space, there's almost no relationship. But when you run it on the density-aware version, you get a much better correlation coefficient. And this is just another data set. This is now a fly-brain data set, a time series data set, where each color represents cells sampled at a different time point. And again, we have the same idea where qualitatively things look very similar, but there are some different shapes that I think are worth interrogating. And again, we have almost no relationship between the original and embedded radii in original t-SNE, and a nice relationship with the density-aware version. And now I'm going to conclude with something that I see far too often unresolved in my code, which is a bunch of to-dos. So first of all, what I'm really looking for is a real-world data set where the density property actually becomes really important, where you can actually see something pop out. So if any of you are running t-SNE on your data sets and not getting great results and think it might be because of density, let me know. And we heard before about UMAP as well. So there's nothing in my algorithm that's specific towards t-SNE. So a good next step would be to actually adapt the density measurements to work well with UMAP. And FitSNE, I don't know if any of you know this algorithm. It's essentially a much, much faster version of t-SNE. All right, thank you.
Hey, thanks for the talk. Quick question. Can you imagine that this addition to either t-SNE or UMAP would be useful for downstream analysis? So most of the time when I use t-SNE is just to look at it, or UMAP, whatever, but I don't use this data to do anything more on it. What do you say about your...
Yeah, I think that's a great question. Essentially, I mean, it's very good, I think, that you don't do that, because I don't think t-SNE is actually very good at... Oops, sorry. Okay. So, so basically that's what I'm hoping for. By incorporating more and more measurements that are preserved in the visualization, I'm hoping that you will be able to use these nice two-dimensional features in downstream analysis. I'm not sure if just this is enough to make it a good tool for downstream analysis, but I do think it helps.
Thank you.
How about MDS, multidimensional scaling? Because that should preserve the density, right? But I mean, have you tried this on the smaller data sets? And it would have probably the same property as you observed with your t-SNE, but probably some other problems?
What is the algorithm you're referring to?
Multidimensional scaling.
Yeah, I'm not familiar with that actually. I'll have to try it. Thanks.
Okay, thanks very much. Okay, so we come to the final